What we're going to move on to look at here are some of the other variables that we can calculate after knowing our glomerular filtration rate, so knowing our GFR. So if we think about our plasma, our renal plasma flow, this is the amount of blood that is flowing through our kidneys per minute. Okay. And when you think about that, that means the amount of blood that's flowing in here into the afferent arteriole that is making its way into the glomerulus. Very different from our GFR. Our GFR, on the other end, is the amount of volume that is filtered per minute. So the GFR is what actually makes its way across this membrane, right? That's going across our filtration membrane. And so when we think about those values, we have about 625 milliliters of plasma flowing into our afferent arterioles, right? So 625 milliliters per minute. But of that, only 125 milliliters per minute ends up being filtered across our filtration membrane. So what that already begins to tell us is that most of that plasma is actually not filtered. Most of that ends up back into the efferent arteriole, back into our capillaries, and especially our peritubular capillaries that are going to be intertwined with the distal parts of the tubules. All right. The other thing we want to compare this to is our systemic capillaries. So our filtration pressure is about 2 milliliters of mercury. And so that gives us a filtration rate of about 3 liters per day. All right, very high in terms of our systemic capillaries. But when we compare this to our kidneys are filtering about 180 liters per day. So just comparing what's happening in our systemic capillaries, filtering 3 liters per day, and what's happening in our kidneys, way higher amounts of volume being filtered per minute, and then eventually per day, right? And then we can also think about how our filtration fraction can be calculated. So our filtration fraction is what percent of our renal plasma flow actually ends up being filtered. So we can take our GFR, divide that by our renal plasma flow, and think about the percentage of our flow that is actually filtered. So just plug in these two values. So our GFR being 125 milliliters per minute. Our renal plasma flow, again, the total amount of plasma that enters our afferent arteriole, that is 625 milliliters per minute. And so that tells us that only 20% of our total renal plasma flow is effectively filtered. The remaining 80% of that ends up in the efferent arteriole and moves its way into the peritubular capillaries. All right, so again, another equation that you should be comfortable calculating, if you have the GFR and the renal plasma flow, you should be able to calculate with very simple math our filtration fraction, the percent of plasma that is actually filtered. We can also calculate our filtered load. This is a third variable that we can deduce given our GFR. So our filtered load is and this is going to be dependent on a specific molecule. So we're going to take a molecule X, for example, and if we know the plasma concentration of molecule X, which is this here, Px, we can multiply that by our GFR, and that will give us our filtered load. So how much of that molecule X is filtered per minute? And so this is only applicable to small molecules that are freely filtered without any impedance. So we're not going to be able to calculate things like a glucose, for example, which shouldn't really be filtered or should be reabsorbed rapidly. Or we can calculate glucose, excuse me, let me apologize here. We're not going to be able to calculate things like proteins or even erythrocytes or even amino acids because they are not being freely filtered. So glucose is, let me just correct myself here. Glucose is freely filtered, and so we can calculate the filtered load of glucose as long as we know its plasma concentration. So it depends on that concentration and the GFR, and we can simply multiply those two variables. So our GFR being at 125 milliliters per minute, our plasma concentration of glucose being 100 milligrams per deciliter. And if we convert that to our consistent variables here, we have 1 milligram per milliliter. And multiplying those two gives us 125 milligrams per minute. So that is our filtered load of glucose, which tells us that 125 milligrams of glucose is filtered every minute, okay, because of our plasma concentration. If we think about our total amount of fluid filtered per day, only about 1.5 liters of urine is excreted, so less than 1%. And more than 99% of that is typically reabsorbed. So of the 180 liters filtered per day, remember we said our GFR is 125 per minute, but 180 liters per day. So most of that ends up being reabsorbed. We want to reclaim most of that, especially in dehydrated states, in states of low glucose, where we need to reclaim amino acids. Most of that ends up being reabsorbed in order to maintain our fluid levels and our fluid balance. Now, another adaptation that we see here is that a small increase in GFR leads to a large increase in volume of fluid that is filtered and that is eventually excreted. So a very small adjustments, a small bump up in our GFR results in a very large volume filtration rate. And we'll talk about some of the regulatory mechanisms that allows for that, but this is a very interesting change that we see here in the kidneys. So GFR is going to be highly regulated with hormones, with extrinsic mechanisms, with some of the cell types and some of the secretions of the kidneys, like our macula densa cells and other cells, but the GFR is highly regulated depending on several factors that we will discuss. So here is comparing our mean arterial pressure, which is, we can think about this as being our overall blood pressure, and we're comparing that to our glomerular filtration rate per minute, milliliters per minute. And so notice in this central zone here, between 80 millimeters of mercury and 180 millimeters of mercury of our mean arterial pressure, notice that our GFR is pretty constant at 125. However, if our blood pressure drops below 80, the kidneys will fine-tune themselves and they will no longer be able to maintain the GFR in the face of that drop in mean arterial pressure, and the GFR will then drop too. The same is true for increases in our blood pressure. If our blood pressure goes above 180, the kidneys again will also not be able to maintain regulation beyond that pressure change, and so our GFR will rise as a response to that beyond 180. So between 80 and 180, the kidneys are able to regulate and keep the GFR at 125, but outside of that, those variables, outside of that value, it will end up resulting in a change in GFR, which is why we want to keep our mean arterial pressure at within homeostasis. Let's say that. We don't want to have very high blood pressures. High blood pressures result in long-term changes to our kidneys that can eventually begin to damage the kidneys. If we raise GFR above 125, especially if it gets way up here at 250, these high pressures can begin to damage the glomerulus, the fenestrated capillaries, the basement membrane, and that can lead to things like nephrosis that I mentioned earlier. The kidneys are not built to sustain these high glomerular filtration levels, and so that can begin to injure the kidney. So we want to keep it between these pressures here, between 80 and 180. Now, there are three intrinsic regulation mechanisms to keep GFR. So again, we're going to talk about how we can keep the GFR at 125, even if we have a change in our mean arterial pressure, as long as it's between that 80 to 180. So the three mechanisms that allow that are one, the myogenic regulation. So the smooth muscle wall in our afferent arteriole, that can detect the pressure of the blood that's coming in. And so a rise in GFR results in a constriction of these cells, which can increase the pressure as well. So high amounts of volume and pressure result in squeezing on those, or sorry, contraction of those arterial, the smooth muscle of those arterial wall cells, and that can increase the GFR, right? The other mechanism, the tubuloglomerular feedback, this has to do with the macula densa cells. And the macula densa cells are a group of cells in between the ascending limb of the loop of Henle and the afferent and efferent arterioles of the glomerulus. So I talked about how the ascending limb comes back into contact with this region of the glomerulus, and that creates this complex. It's going to have your macula densa cells. It also has your extraglomerular mesangial cells, some additional cells that can also help with the fine tuning and bring about corrective mechanisms. And then it also has some granular cells that are found along the outer surface of our afferent arteriole. So these cells are going to secrete certain paracrine factors, and paracrine just means that they're like hormones, but they're not traveling in the blood. They're kind of cell-to-cell signaling factors that are released in response to increased flow. So if we have a lot of flow coming out here in the distal part of the tubule, specifically as it crosses through the ascending limb, that can trigger a response to the cells here to help the smooth muscle of the arterioles contract in response to these paracrine signals. And so that too can cause a contraction that increases GFR in the face of increasing flow coming through the ascending limb. And then the third mechanism is the mesangial cell contraction. So mesangial cells are cells that are surrounding the capillaries. They're found in between the capillaries of the Bowman's capsule. And when they constrict or contract, they can also help to increase that pressure and increase that GFR. All right, so here's what that complex looks like on histology. It's between the distal part of the tubule here and the glomerulus, especially the afferent and efferent arterioles of the glomerulus. And it is comprised of the macula densa cells of the ascending loop, the extraglomerular mesangial cells. So there are two types of mesangial cells. Some that are in between the glomerulus, which we call mesangial, and then some that are extraglomerular. They're outside of the glomerulus, but they're also considered mesangial cells, and they can constrict and increase the pressure on the glomerulus, which is a response to regulate our GFR. The third structure that is forming our JG complex is the granular cells of the afferent arteriole. Okay. Let's not get into this today. Let's go ahead and end here for today. And then when we meet on Monday, we'll go through these mechanisms in more detail and talk about the different steps, the feedback mechanisms, and how we result in the change in GFR.